In this video, we will describe the method for producing dihaploids from cultivated potato. Cultivated potato is a tetraploid. It has 4 copies of each chromosome. Dihaploid potatoes are diploid. They have 2 copies of each chromosome. Dihaploids may be useful for genetics research and for breeding of diploid potatoes. The process of dihaploid extraction is quite simple. A flower on a tetraploid potato is pollinated with pollen from a special diploid line. The resulting dihaploid seedlings are derived exclusively from the female. The female parent can be any potato variety or breeding line, one that has valuable traits. The only requirement for the female parent is that it be female fertile. Only a few lines will produce dihaploids when used as the male parent. We have used IVP 101. IVP 101 flowers are somewhat reluctant to shed pollen, so we often pick them, let them dry for one day, and then buzz out the pollen using a modified electric toothbrush into a gel capsule as shown here. Another way to encourage pollen release from IVP 101 anthers is to harvest fresh flowers, cut off the tips of the anthers with a scissors, wait a day for the flowers to dry, and then buzz them to release the pollen. The first step in the process is producing flowers. This can be done either in a greenhouse or in a field. In the greenhouse, it is helpful to keep temperatures cool, lights bright, and to fertilize heavily, as these practices promote fertility. It is also good to grow the plants in relatively large pots and to space them out so that they are not crowded. Alternatively, one can take advantage of the very large number of unwanted flowers produced in commercial potato production fields, and one can collect a very large number of flowering stalks in a very short time. These flowering stalks can be brought back to a greenhouse and placed into bottles or tubs containing water. There, they can be used for pollinations, just as you would use flowers on greenhouse-grown plants.
The variety is pike, and I'm crossing it with our free pollinator, which is IVP 101. Previously on this plant, I did 4 pollinations on September 28th, 3 on the 30th, 4 on the 2nd, and then I will do some more today. You can do multiple pollinations on a plant as long as you keep up on it and don't let flowers get too old. So here, for example, this where there's a berry forming, the biggest berry, that was done on the 28th. This next one was done on the 30th. And then these smaller ones on top were done October 2nd. So now today on the 5th, I'm going to emasculate those that are ready today. I can do buds. And the rule of thumb is if you start to see the petal color, that's mature enough to emasculate. So I go in and I'm removing sepals, petals, and the anthers. So emasculation is demaling the flower. So I remove the male portion, just leaving the female portion of the flower. So I did that one. I have this one as a rule of thumb. If I do not see the anthers open, I can use it to pollinate. And in the greenhouse, there isn't a lot of movement or vectors to pollinate. So I just go around the flower, twisting it this direction and that direction. So for today, I have 3 on this inflorescence. This one I'll wait in a couple of days. I can do a few more. Again, that one actually looks like the anthers are open, so I'm going to remove it. And that one looks okay. So again, there I don't have to take the petals off. Here I can do this one. And I just use my fingernails to do this. Here I'll do these 2. And some flowers are a little more brittle than others, so they're like that. The style just broke, so I'm going to remove it. Now I'm going to use my pollen that I collected, and we use these gel caps. So this is the IVP 101 pollen. So basically all I do is I rub the side of the capsule up against the stigma. And if you can see there is pollen on there. So you can see that with the naked eye. And I will just do, that's 2 and 3 on that inflorescence. And then on this other one, I have 2 to do. Just be careful with the inflorescence that you're not stressing it too much and breaking things. And you can even repollinate something you did a couple days prior in case there's different levels of maturity of the eggs in the ovary. So that's all you do for pollinating. If things are successful, you'll get a berry that will form. So
After the berries form, we'll allow them to develop on the plant far enough so that they can be handled gently. Then we'll enclose them in a nylon bag and let them develop further, typically for about 4 weeks after pollination. The berries can then be harvested.
So in here I have berries that came off of a clone. You can dump them all together. One way you can do this is by just cutting the berry open if you have limited berries, and then squeezing the seeds out on a paper towel. And then you can let that dry and then bag them up later. The other way I like doing this is berries are left on the plant for 4 weeks, then I pick them and let them ripen for another 4 weeks so they're nice and soft. And then all I do is squeeze them inside a beaker of water. Heavy seeds fall to the bottom. Seeds that don't have an embryo or are immature would float to the top. I throw the pulp out. I can decant off any pulp. You can rinse it off. I'll let it sit. This one is sitting. I have my seeds in here. And then what I typically do is I dump them in a little nylon bag. Get them all. Then I typically rinse these off with some lukewarm water if there's a lot of mucus still on the seeds, because that'll extend their dormancy. Finish it with cold water. Put my tag on the bag, and then I'll let that sit on a greenhouse bench for them to dry out. One other thing I'm going to point out is we're hoping that these are not self-pollinated, but if they would be, there'd be a lot more seeds per berry. If that is the case, I would keep those separate or throw them away. Typically, I've been getting on average about 15 or under seeds per berry, knowing that it's not a self.
The pollinator that we use contains a seed spot marker. If the male contributes its genetic information to progeny, the embryo will have a small purple spot that is visible through the seed coat. Seeds that show this small purple spot are discarded as they are likely to be tetraploids. Producing dihaploids reveals many deleterious alleles in progeny, and that can lead to a wide range of phenotypes, most of which cause plants to be less vigorous or less fertile than their tetraploid parent. There are several ways to confirm the ploidy of putative dihaploids. One way is to use genotyping with molecular markers, such as single nucleotide polymorphisms, or SNPs. This will distinguish between tetraploid and diploid, and will also give additional information with regard to duplication or deletion of smaller segments of the genome. Another method is flow cytometry, which directly measures the amount of nucleic acids within the nuclei. A third way is to cross putative dihaploids with a diploid male. We have used the diploid M6 most frequently. M6 has very high male fertility and rarely produces 2N pollen that would allow it to cross successfully with a tetraploid potato. Finally, one can count the number of chloroplasts per guard cell. In tetraploid potatoes, we have between 9 and 14 chloroplasts per guard cell, depending on how the plants are grown. In diploid potatoes, we have about half that amount, between 4 and 8 chloroplasts per guard cell. Guard cells are most clearly visible in epidermal peels. Let's see how to prepare those.
I'd like to show you how we make an epidermal peel. What I do is take the terminal leaflet from the first fully expanded leaf. I pluck off the terminal leaflet, put some water on a microscope slide, then I take the leaf, trying to tear it in a way that gives me a single layer of epidermal cells. Like that. Then what I will do is to just so I've removed the rest of the terminal leaflet. Setting that on some water, the microscope slide. Then I lower the cover slip. And move to the microscope to make my examinations.
The success rate for dihaploid extraction is quite low, and many of the dihaploids produced have very low fertility. So why do we do it? We do it because the dihaploids we generate now will be the foundation of the next generation of potato, which we are calling Potato 2.0. Diploid potato varieties produced from these founding dihaploids.